Hello, my name is Marom Bikson, and I'm going to talk about the history of the creation of high-definition transcranial direct current stimulation, or HD-tDCS, which is also a basic introduction to how HD-tDCS, in fact, works. Okay, so there's different types of neuromodulation technology. The name usually tells you how the energy gets in, whether it's by transcranial magnetic induction, like with TMS, or with electrodes implanted deep in the brain, like deep brain stimulation, or transcranially, as in the case of transcranial electrical stimulation or transcranial direct current stimulation. And just I would note that, whereas some techniques require a device to be implanted inside your body, others are associated with enough risk or cost that there would be something that would only be in the hospital. There are other forms of neuromodulation that are truly wearable, they're battery powered, and transcranial direct current stimulation would fall into that category. And it's generally thought that as you move sort of from one side here to the other, you get a more targeted intervention, right? For example, by implanting an electrode deep in someone's brain or in their spinal cord. But then also there'd be an associated risk, as it with any type of surgery, or perhaps risks associated with seizures and so on. So this is sort of the inherent trade-off. And really, I think the big picture as far as high-definition TDCS goes is really asking, well, is the wearable transcranial electrical stimulation technology truly limited in its targeting, or perhaps not? And obviously I'm going to suggest that perhaps not. So starting though with conventional transcranial direct current stimulation, this is what it involves. You have a function generator, essentially, that is producing a direct current, like a 9-volt battery, with an intensity of a few milliamps, like a 9-volt battery. It is connected to the head via 2 wires, and those wires are connected to sponges that are then placed on the scalp. And now you need to imagine that there is a circuit of current flow traveling down the red wire, heading to the brain, where it hits that first sponge, passing through the head, being collected at that second sponge, and then being brought in. So this loop is required for an electrical circuit. The sponge that is connected to the positive terminal of the function generator is called the anode, and the sponge that is connected to the negative terminal pole of the function generator or stimulator is called the cathode. And of course, to get from the anode electrode to the cathode electrode, well, there's a head in the middle, and inside the head is the brain. And so by pushing current around in the circuit and by having the head as an essential part of the circuit, we produce current through the skin, through the skull, hence transcranial, and then we stimulate the brain with direct current. So you see how the name tells you a lot about what it does already. And again, this is conventional TDCS, not high definition, using large sponges. And a lot of our work tries to address how these technologies work using computational modeling. And I have a video that discusses in more detail the use of computational modeling, as well as all these terms like anode and cathode. But for here, we just need to understand that with conventional TDCS, you have, again, these 2 relatively large electrodes, a few centimeters by a few centimeters, and they're placed somewhere across the head. And according to computational models that we developed, these were the first realistic MRI-derived models of current flow through the entire head, we guessed that when you do this, the current flow is not targeted. In fact, it flows all the way from one electrode to the other electrode, thereby influencing not just everything under these 2 relatively large electrodes, right, but also everything between them. So that's that current flow pattern that you see, where the current is really washing across the entire head. And in fact, sometimes the peak is not even under the electrodes, but somewhere between them. And this was the result of a computer simulation first published around 2009. And this led us to suggest an alternative, where instead of doing conventional TDCS, we will do something called high definition TDCS. And in this case, you can see that there are more electrodes placed on the head. They're smaller, they're in a particular pattern. And when we ran these simulations, we predicted that when you do high definition TDCS, as opposed to conventional TDCS, you will produce focal brain current flow. Specifically, if you have a bunch of electrodes, the current flow will be restricted sort of by the ring defined by these electrodes. Again, this was shown first in a computer simulation, also published in the same paper in 2009. So why is it working that way? going back to conventional TDCS, you have this large anode and you have this large cathode, and they're placed here on top of this sort of cartoon scalp, and then skull, and then cortex underneath it, and it's connected to a TDCS device. And as I mentioned, current is pushed out of the TDCS device into the anode and therefore into the cortex, and then it is collected out of the brain at the cathode. And so that creates the loop. And now we can imagine you have a cell in the cortex under the anode, in a red dot. You have a cell under the cathode electrode, also in the cortex, in the blue dot. And so current is passing along these cells, right? They're being affected by the current, and this is where we get the stimulation. Now, it turns out that the kind of stimulation you see under the anode and under the cathode are very different. Under the anode, we have something that we sometimes refer to as a nodal TDCS, soma depolarizing, also referred to as excitatory TDCS. Under the cathode, where we still have current flow, but it is now in the opposite direction, we produce an opposite profile of polarization, cathodal TDCS, leading to somatic hyperpolarization and a decrease in excitability. And again, in a separate YouTube video, I talk about how this polarization arises and how even if this polarization is very weak, it can still lead to changes in brain function. But for now, we just need to understand that there are two different forms. And because we have two electrodes, we always need to contend with the fact that we're producing both the nodal excitatory variant and the cathodal inhibitory variant, right? TDCS has an anode and a cathode, and therefore always a nodal and cathodal happening. Now, there aren't just two cells, there are many cells. I'm illustrating here these with many dots, a bunch of red cells getting a nodal, a bunch of blue cells getting cathodal stimulation, but the current doesn't actually flow in these sort of simple straight lines. As I suggested, it has to flow in and across the entire brain and then be collected by the other electrode, and it can actually spread out a little bit along the way as well. So the polarization profile looks something like this, right? Where really you're getting a vast tract of the cortex spanning not just the areas under the electrode, but between them, all around them. And so this is why with conventional TDCS, we predicted you would produce a diffuse, non-focal form of brain stimulation. Two large electrodes, conventional TDCS, leads to diffuse current flow, and so diffuse brain stimulation. What if you make the electrodes smaller? Would that result in focal current flow? Well, according to our intuition and also our modeling analysis, the answer is no. Because even if you make them smaller, the current still needs to get from one to the other. The current can't enter the brain under the anode, right? Then disappear and then suddenly reappear under the cathode and be sucked out. The current must be continuous. The circuit is continuous. And so the entire area between these electrodes and also a little bit around is still being stimulated. Getting too small electrodes does not produce focal stimulation. Now, we call these small electrodes high definition or HD electrodes. And I'll be coming back to that, but I just want to say just having small electrodes doesn't lead to focal stimulation, okay? So what do you do instead? Well, one thing you could do is you can move these electrodes closer to one another. Now, one of the reasons you can do that now is because they're smaller, right? When they're two big pads, how close can they get? But when they're two smaller ones, you can move them closer together. And this does produce focal stimulation, right? And again, I have an additional video where we talk all about how you get targeting and how you customize even two electrodes to produce the kind of outcomes that you want. For now, I think it makes intuitive sense. Two small electrodes at a distance, not focal. Two small electrodes close to each other. That does produce focality, but you still have this two-way street, right? Where under the anode, we're producing a nodal stimulation, excitatory, and under the cathode, we're producing inhibitory stimulation, though it is now focal. All right, two electrodes, still two regions of influence. The next step in the creation of high-definition TDCS was thinking about having additional electrodes. So in this case, what you're seeing is that there's one center anode electrode with current being pushed in, and two outer cathode electrodes with the current being collected out. And if you, so we might call these two cathode electrodes the return electrodes. And because there are two of them, they are weaker. If you can imagine that we're pushing in, let's say, 1 milliamp through the anode, The 1 milliamp has to be collected in totality by the cathode, but they split the work. One sucks up half a milliamp, and the other one sucks up half a milliamp. And so now we have sort of a symmetry where we have a stronger anodal effect and a weaker cathodal effect. And on the head itself, we can even expand this to not just 2 cathodes, but 4 cathodes. And so in this case, if we're pushing in 1 milliamp, each cathode would be collecting just a quarter of a milliamp. So you see how the cathodes have been made weaker. We have 1 center anode, and now we have 4 collector cathodes. And this is where we get 4x1 high definition TDCS. It's TDCS because we're pushing in direct current through the skull. It is high definition because we're using high definition electrodes, and it's 4x1 because the way we have arranged them is 4 surrounding 1. So this is what 4x1 will look like on the head. Again, rendering of my head, you have 1 center anode, 4 surrounding cathodes. Now, how would you connect that to a device? You can make a custom stimulator, or in fact, you can use that with a traditional stimulator by taking the anode wire, running it to the center electrode, and then taking the cathode wire and splicing it into 4 wires, and then taking all those 4 wires and connecting them each to each of the 4 electrodes, or you could actively split it. So this is how you would just implement the 4x1. And when we simulated this idea, what we found was something interesting. Not only was the current more focal, but it was also more unidirectional. The red here means current flowing in. Let's call it the anodal current. The blue means current flowing out, whereas the green means no current, right? So it's a little different than the colors I showed you before. What the models were telling us is by doing 4x1, we could produce a dominantly, 4x1 with an anode in the middle, produced a dominantly anodal current flow with a much weaker and distributed cathodal current flow. So showing us not only that we can produce transcranial focality, but now we have a more unidirectional form of polarization. Okay, now the key thing to understand here is that when you to put the 4x1 on the head, the area that is being stimulated is not the size of the middle electrode. That was never the case. It is the size of the ring, right? So the size of the ring defines how far the current can spread, and therefore how focal we're going to be. And so the region of the brain that is influenced is sort of the projection under the ring. If you make the ring smaller, then you will make the stimulation more focal and you will influence less part of the brain. Now there's some loss of intensity because of shunting at the scalp between the electrodes, but you can compensate for that by just applying more current, right? So we do lose a little bit of intensity as we become more focal, but you can compensate for it. And this is something that, again, we've addressed systematically and quantitatively in computational models. This is a simulation of 3 subjects looking at different sizes of ring, going all the way from 3, 4, 6, 7 centimeter radius. And you can see the size of the brain that you influence is the size of the ring. You can also see this is very robust across individuals, right? So we set the ring, you put it over a part of the brain, and regardless of the individual, you're going to be driving that current in focally. You can also see here that there's a little bit of loss in intensity as you become more and more focal. But again, this is for a fixed current and you can compensate for it. But once again, there doesn't seem to be a hard limit on how focal you can be with transcranial electrical stimulation when using the 4x1 high definition TDCS montage. And we also realized that using this approach, you can target any cortical region. So here we have it over one part of the brain, but you want to target, you stimulate parietal, you want to stimulate occipital, you want to stimulate frontal. It's as simple as moving the 4x1 over the part of the brain that you want to influence, and now the current again will be restricted under it. Now it turns out you have many potential places you can put these electrodes. One way to figure out where to place them is by using the EEG 1020 or 1010 system. It's just a coordinate system, C3, C4, that allows you to place them on. And so when you see high definition being applied, it will often be prescribed according to where you place it on the EEG 1010 system. But whereas the EEG 10 system may have 64 positions, you only need electrodes in 5 for the high definition TDCS. And this is what it will look like on the head with all possible positions, but you're only selecting 5 of those in order to draw up your high definition electrodes. Okay, so we developed this now a while ago, and there was just one problem. As I was going around speaking about it, no one believed what I was saying. And they had very good reason not to believe it, because these were computational models. Everything I've shown you so far is a computer prediction, and people were a little bit skeptical, right? They'd grown up learning that transcranial magnetic stimulation is focal and transcranial direct current stimulation cannot be, and here we are suggesting that that wasn't the case at all. And so correctly, there was a valid pushback against the reliability of these models. And so we and others now have done multiple forms of validation experimentally. Let me show you just a few examples. This was an experiment. It was a result that was published in 2013. Now what you're seeing here is the 4x1 montage simulation on my head. In the middle row, you can see that the 4x1 is over the motor cortex, the part of the body that makes your arms move, right? In the top, the 4x1 has been displaced forward, and in the bottom, the 4x1 has been displaced backward. Now look what happens. When you displace it forward or backward, you still get current into the brain, but not to the motor cortex, because it's focal, right? Because it's focal, when you move it, you're now focal somewhere else. And so the prediction is that when you displace this electrode to these different positions, you will either get current into the motor cortex or you will not. Now, when you normally do TDCS on people, or high definition TDCS, other than feeling some tingling and skin redness, there isn't an overt physiological response. It's not like TMS. When you do TMS, the intensity is so high that you will produce an involuntary finger twitch. With TDCS, you don't, unless you increase the current level. So after positioning the electrodes in these 3 positions, over motor cortex, anterior or forward or posterior or backward, we now applied stimulation, but not with 1 milliamp, but with 1,000 milliamps, a very high intensity shock. And when you do that, the amount of intensity that gets into the brain is enough to make your finger twitch. And you can measure that twitch via a motor evoked potential or an MEP. So the blips that you're seeing here, and again, I'll refer you to the paper below for more details, but the blips that you're seeing here are telling you whether the finger twitched or not. When we did 4x1 over the motor cortex, we got a finger twitch. When we moved it anterior or posterior, the finger twitch went away. And this is consistent with the model predictions of focality. Now in the same paper, by the way, with more resolution that I'm showing you here, we compared the focality of 4x1 transcranial electrical stimulation, right? This is with 1,000 milliamps, with TMS. And at least at the level of resolution that we looked at in this paper, we couldn't decide which one was more or less focal. But the evidence here was very clear. 4x1 can deliver the current focally to the head. Years later, we continued to work on, we continuously worked on validation, including this paper that was published in eLife in 2017. Now, in this case, you had patients who were candidates for epilepsy surgery, and for that reason, they had many, many electrodes implanted inside their head to measure where their seizures were coming from. But those same electrodes could measure current flow as we applied transcranial direct current or transcranial alternating current stimulation to these subjects. I'll refer you to the paper for all the details, but bottom line is, on a subject-by-subject model basis, montage basis, we found that the models were correct. The current flow patterns, which were diffuse, because this was conventional TDCS, that were predicted by the models, quantitatively matched the experimental recordings with these depth electrodes. And so this was one of, I think now, 20 different validation efforts showing that these models were right. And if the models are right, it also means that high definition does what we're predicting it to do. Now, up to now, I've really focused on the 4x1 HD-tDCS montage. But as I mentioned, you have many possible places that you can place the electrodes. And so we developed, and others have done similar work, algorithms, right, that allow you to say, okay, I want to go to a particular brain region. I want to stimulate this brain region. I have some constraints. Maybe my stimulator only has 8 channels. Maybe I don't want to do more than 1 milliamp per channel, whatever your constraints are. And then the software will automatically give you a prescription of where you should push the electrodes and how much current you should put into each one, because if you have a multi-channel stimulator, you can independently control the current at each one. And this is actually a closed form solution subject to constraints. And one of those most critical constraints that we realized is that there is a trade-off between maximum intensity, so how strong you stimulate a brain region, and maximum targeting, meaning how focal you are. This is not specific to high definition TDCS. This is for all forms of neuromodulation. You get this sort of trade-off. It exists here as well, and you must factor it in when you are designing interventions, again, as described in this paper. Okay, there was just one other problem. In 2009, which is when we first published these computational models, remember with the conventional TDCS and then the focal TDCS, we needed small electrodes to get it to work. You cannot use sponges. And the small electrodes didn't exist at that time. And moreover, people said that it wouldn't be possible to make these small electrodes. Now, why did they say it wouldn't be possible? Because they said it would hurt. The current density would be too much. Current density at the electrode, average current density, is defined as the current that you're putting in divided by the area of the electrode. So if you might be putting in 2 milliamps of current, let's say. The electrode is 5x7, so it's 35 centimeters squared. And so the current density is 2 milliamps per 35 centimeters squared. And people realize that as you start to make these electrodes smaller and smaller, if you kept the current the same, but made the area smaller, the current density increases, right? And again, I talk elsewhere about current versus current density and so on, but that is the basic idea here. And so people said, well, as you make the current density higher and higher, it's going to get more and more uncomfortable. And that's true with sponges. And therefore, you can't make small electrodes. And if you can't make small electrodes, you don't have high definition, and you can't do anything else I've shown you. So a high definition electrode is ultimately defined as a small electrode. In our cases, around 1 centimeter in diameter. And again, if you keep the current the same, but push it through but try to push it through that smaller electrode, the current density goes up, and people were telling us it couldn't be done. Well, maybe we didn't know better, so we tried to make it work. And so the discovery of high definition electrodes, in the title of the paper, was published. The work was done a little bit before 2010. It was published in 2010. And these are the results of that paper. And I'll refer you to the paper for all the details. But what we learned in this paper and have since applied to a lot of electrode design is that there's only 3 rules that a non-invasive electrical stimulation electrode needs to meet, 3 standards in order to be good. That's it. 1, when you stimulate through the electrode, right, with the current that you want, it doesn't hurt. And that you can find out just by asking people, right? Second, doesn't damage the skin. And that you can find out by looking at the skin right after, 24 hours later. The third aspect of it is the resistance can't be too high, and this is a technical issue related to the fact that we're using current-based stimulation, and if the resistance is too high, then the voltage is too high. If you meet these 3 criteria, you're good. And by trying out different electrode materials, different electrode shapes, different gel materials, we were able to ultimately, after a lot of work, meet these 3 criteria. That was the high definition electrodes, and that actually led to a startup called Soterix Medical. So Soterix Medical is a company that I co-founded that makes high definition TDCS, but at the core of the enabling technology is actually the high definition electrodes. Right? And so this is what a modern high definition system looks like, made by Soterix Medical. You have a cap, and the cap is fitted with high definition electrodes. Could be 2, could be 5, could be the full complement that you're seeing here. You have a much more sophisticated function generator, right? This may have multiple waveforms, multiple current control channels, and you have a piece of software that is running, that is helping you to decide where to place them, even on a subject-by-subject specific basis. So now I think the rationale for all this becomes a little bit clearer. I also want to mention one nice feature about high definition is, while it's not the same as an EEG electrode, their form factor is very similar to an EEG electrode. And so what you can do is in a single cap, right, you can combine high definition electrodes, in this case shown as the bigger circles, with recording electrodes. right? So now it's very convenient to do brain reading with EEG and brain stimulation at the same time. In some cases, you can even use the same electrode. And I do want to mention that a few years ago, we published an algorithm based on a principle called reciprocity that described how you could record a brain signal with the EEG and in a closed form way process that in order to then stimulate the brain optimally to activate the same parts of the brain that generated the signal, right? And again, I'll refer you to the paper for all those details, but also these systems now exist. One more really important experiment. The original discovery of the modern discovery of transcranial direct current stimulation was by Nietzsche and Paulus, and they did a very classic experiment where they applied TDCS with conventional sponges, which I mentioned doesn't produce like an overt response, and then they used TMS muscle twitches to assess how is the TDCS changing brain excitability as can be detected by doing TMS muscle twitches. I'll refer you to that paper if you're not familiar with that technique for more detail. It was very important that they did this work because they were, again, among the originators of TDCS, so it was very meaningful for them to then compare it to high definition TDCS. And what they report in this paper is very interesting. It turned out that both the time course and the intensity of excitability changes produced by TDCS versus high definition TDCS are very different. They reported that 2 milliamp of just 10 minutes of 4x1 HD-tDCS led to over 2 hours of excitability changes, right? Which was much longer than when conventional TDCS. I think the main thing I want you to take away from this slide is that the way high definition TDCS and the way conventional TDCS change the brain may be rather different. When you're focal versus diffuse, you can produce very different dynamical responses as far as the brain response. This is rather complicated, but this work and now 100 human trials using high definition TDCS is starting to teach us how. So I hope that this has been educational and clear. I'll try to explain how high definition TDCS was created, but in so doing, what are the essential features of TDCS. And again, I would refer you to other videos that I've made where I really talk more about all the different ways you can customize HD-tDCS to guide current to different parts of the brain, as well as videos where I talk about why does direct current even change the brain to begin with. Thanks for your attention.